with session 5. Thank you all for attending this session in this virtual SCA ARCA Global Conference. I am Dr. Pedroso from Brazil, and it is my great pleasure to be the chair of this session 5, Existing ARCA Cohorts. This is really a wonderful opportunity for ARCA experts to introduce the ataxia centers of their respective continents and for future collaboration. I'd like to remind the speakers to respect the presentation time, 12 minutes for presentation and 3 minutes for questions, and all presenters will be available for questions at the end of the session. You may send your questions through the Q&A chat. And to begin, I would like to introduce Professor Matthis Sinovsky, who is the ARCA Global Chair, Professor of Neurology in Tübingen, Germany, and also the founder of the ARCA Global, that will present the topic ARCAs in Europe, Prepare. Thank you, Professor Matthis. Your microphone.
Sorry, thank you very much, Jose Luis, for the nice introduction. I would like to start this session. Each of the talks which are going to see has pretty much the same style. Presentator is going to show is in a structured way how recessive ataxias are captured at the respective continent, how many are there, which genotypes, and what data quality is available of those patients in the respective continent. And I would like to start with Europe, and Alexandra Dew is then taking on and giving a second look on Europe. I'm going to talk about are mainly captured in the ARCA registry. And what is the ARCA registry? The ARCA registry is a registry which captures data from a whole different fields of consortium. It has developed nicely since 2012 when it was funded by the German DZNE, German Center for Neurodegenerative Diseases. Since then, the Prepared Consortium is contributing data, and the Ataxia Study Group is contributing data to this. And with all those different networks contributing to this registry, we are now at 28 centers contributing to this registry. And if you have a look at the catchment area, it's still quite European. So I think it captures quite a few of the sites from Europe, but also, for example, some from Canada, mainly contributing ARCA patients, but also, for example, from Tunisia, Turkey, and even New Zealand. Richard was mocking at me that I always forget New Zealand. So Richard, there's New Zealand on the map. So it's getting more and more global. So it's not only a European perspective, not only European data I'm going to present here. How is this registry structured? This registry is a web-based registry, so you can enter it from any laptop, any computer, anywhere in the world. It's a multi-center and it's GCP compliant, which is important if we now talk about trial readiness. It is used, as I said, by those different networks, and this is why we keep increasing the numbers in this registry. But what is really nice for the users working with this registry is two things. First of all, it has the same CRF forms than, for example, other recessive ataxia registries like the Prospex registry, which captures RSAX and SPG7 now, it's now starting, or for example, the Treat HSP registry, which has to focus on HSP. And now giving the overlap between recessive ataxias and HSPs, this allows us to go to do common downloads and do common data analysis in this overlapping field of spastic ataxias. And this is why it's so helpful to have the same data elements and to have the same CRFs. Also, the same or very similar front ends and very similar CRFs are by the Sportax Natural History, by the SCA registry, and by the ESME. Of course, SCA3 probably won't do combined analysis with the recessive ataxias for ESME, but for example, for Sportax now, if we find the RFC1 patients, this will allow us to run joint analysis by having the same type of CRFs. So I think if using this ARCA registry, one should have those benefits in mind. And for the users, it's friendly because they know how to operate on the respective front end, and they know how to click it and how to solve the problems on when going through the CRFs. This ARCA registry, which I've been showing you, is linked to an ARCA biomaterial database, which is hosted by the University of Tübingen, and we are now establishing an API linking this ARCA registries to a REDCap-based biomaterial database. And the numbers I have here are already 2 years old, but we have more than 600 blood samples already of the ARCA patients. And what we use them for example, is the DNA from those blood samples we just use for whole exome and whole genome sequencing of those patients, ARCA patients who are still unsolved. And those data are then directly fed into Genesis, and you're going to hear about Genesis later tonight. So that's the ARCA registry being linked to the biomaterial database. And what we're going to add to the next years, we're going to link it to a second REDCap database, or ARCA study database, where we can use this second database to extract the readout features from other non-clinical outcome measures like MRIs, Q-motor, APDM, and upload those readouts and then link it to the registry. This meaning also allowing us to have non-clinical data registry. So that is the structure of the registries we are working with. How many patients do we have captured? So far, we have captured a good 1,000 patients and yielding more than 1,500 visits. You can see most of them have still only one visit, but you see we have even some, I think a few patients, 2 or 3, which are now in visit number 8. So this means we already, the first patients are really having long longitudinal registry captured data. And you will see this in the papers coming out later, that we can now really use already those longitudinal data for first analysis. So 1,000 patients, 1,500 visits. What are the patients we are capturing? First of all, 46% of the ARCA patients have an identified genetic course. Still more than 50% are genetically unsolved. And those are the ones we are working with. As I mentioned, the goal is to have of each of those patients either a whole exome, whole genome data set. So we want any patient in this data set to be either genetically solved or have a genome data set in Genesis. For those who are solved, what is the frequency of the genes? And the topmost gene is ARCA. That's certainly a recruitment bias, given the strong contribution by our Canadian friends from Quebec. The second most common, that is not surprising, is Friedreich, then SPG7. So this really tells us that SPG7, we really have to be on the list now for the recessive ataxias, more than, for example, AT, which has long been thought to be the second most common recessive ataxias. SPG7 is likely more common. And then you see it just comes down, and the numbers get smaller. And I think in the others, there's still quite a number of RFC patients still in there. So I think this is going to be maybe number 3 or 4. So this is the distribution of the genotypes you're seeing. What is the distribution of the assessments? We have a cross-sectional, so one-time assessment from 1,000 patients. And you see in blue, those patients where we have 2 or more, so longitudinal assessments. So we have 200 patients with at least 2 longitudinal assessments. Basic clinical features are from almost all where we have a cross-sectional assessment, from all we have longitudinal ones. From all we have SARA. From most of them, we still have the INUS. We have ADTA from a good share of those patients, and we have at least a summary description of the MRI. So this pretty much corresponds to most of the main features which Thomas Clockhetter has pointed out in the clinical outcomes working group. But we are now also adding further items like the FARS RDL to capture more items and make sure that they are captured on a regular basis. So those are the data we can work with. What are the exemplary studies and results we have received so far based on the registry? So far, we have analyzed the data of the COQ8A, so ADCK3 ataxia, and we have used the registry to do cross-sectional longitudinal analysis. This work was published by Andreas Traschwitz in Annals of Neurology. We have one study which is now under revision with Neurology on RFC1, and the second study also under review by Friedemann Benya from Lukas Scholz group on POL-G. So those are 3 just exemplary recessive ataxias where we have already been able to use nicely the registry. A new, more in-depth study we're now starting using this registry is a prospective RFC1 natural history study, and many of you in the audience are already contributing to this study, and that's an invitation for you to use this study, use this registry to run this study together with us. we wish to go into more depth in data analysis, and we are not very far with that as of yet. But just to give you a sketch, it's really a work in progress. Sophie, don't kill me for those data as of yet. But what we really want to do is we want to compare, giving this rich data set, which we would like to have, we want to compare the evolution of the recessive ataxias, and for example, map the evolution of RSACs versus SPG7 versus AOA2, and compare the disease trajectories between ARCA genotypes. But for those genotypes where we have a substantial share of patients, we also would like to go within genotypes and, for example, compare the RSACs evolution of Quebec RSACs patients versus non-Quebec RSACs patients. And what you can see here, appreciate on the right side, is that, for example, the Quebec RSACs patients do seems to start slower, but then ramp off. So we have to go into depths of those studies, but those might be exemplary analysis which we are running on this registry. So with this, I would like to finish my talk, and just there's an open invitation to anybody around the world to contribute to this registry. It's accessible from anywhere in the world. It's web-based, and it's used already, for example, by New Zealand or Iran, so you can use it from everywhere in the world. And I want to thank Andreas Traschitz from my team and Doreen Müller from my team, who are really the two persons from my team who are leading the registry and doing all the main work on this. Thank you very much.
Thank you, Professor Matthis Sinovsky, for this very clear presentation, wonderful data from Europe about ARCA. And I have no question from Q&A, but if you allow me, I would like to firstly to congratulate you after meet you in last Movement Disorders Congress. I was very excited about your project, and there are some points that I'd like to point out. For example, I saw in your data that RSACs is more common than Friedreich ataxia in your ARCA data. How do you explain these findings?
Yeah, so I don't think the number of RSACs in this registry, that is not a representative figure. That is a recruitment bias, which results from the fact that we have those strong Canadian friends, Bernard Brais and Cynthia Gagnon from Quebec. And this leads to, and they have strong partners, our consortia, and this leads certainly to overrepresentation of RSACs. Vice versa, the Friedreich number is quite small, probably it's an underestimate, given that most of the patients in Europe are still being captured by the effects registries. That is a strong Friedreich ataxia registry in Europe. And so I think we have to account for that most Friedreich patients will not be double entried. So that is an underestimate in terms of FA.
Perfect, thank you. I have one last question for related to your presentation. Vikram Shakotai asked, does the registry have information on biological validation of VUS in SACs or other genes?
As of yet, this ARCA registry does not have this yet, but what we have now a distinct prospective natural history study on this fully dedicated to RSACs and SPG7, which is run by the so-called PROSPAX consortium. And one of the main work packages of this PROSPAX consortium, led by Nasli Bajak from Istanbul, is to work up all VUS in SACs on a functional level, and this information will then be fed back in the PROSPAX registry. And this PROSPAX registry has the same front end than the ARCA registry, so they can also talk to each other. So no, it's not in the ARCA registry, but it's going to be in the PROSPAX registry, and they can talk to each other.
Thanks, Matthis, for the wonderful project. I think this will change our history in the ARCA patients. Thank you very much. And now I would like to introduce Professor Alexandra Dew, who is Professor of Neurology in Sorbonne University, Paris, France. Professor Dürr has a relevant contribution in the study of ataxias and SPGs in the world, and is one of the coordinators of the SPATAX, and she'll present the topic ARCAs in Europe, SPATAX. Please, Professor Alexandra Dew.
Thank you. Can you hear me? And can you see my slides?
Yes.
Yes. Can you? Okay,
Yes.
great. Thanks. So yeah, thank you very much, Matthis, for inviting me. And you gave us a real strict outlay how things have to happen. So I'm not so sure if I give all the points, but I will try to take you through on ARCA, also from Europe, but from an older network, which is called SPATAX, and which I explained to you because why it's called like this, even if you perhaps already had some of the introduction on this network that exists for a while now. So your first question was, beyond your own sun, do you have a larger countrywide national and continental network, and what's the geographic catchment? That's what you asked us to do. And yes, we have. And why it's called SPATAX? It's because with Chantal Talaxen from Oslo in 2000, we were coming across quite a lot of patients sharing both spastic paraplegia and ataxia, either in the same patient or in the same subset of families we met. And so we decided to do a network that would profit from expertise in both groups of research areas, cerebral ataxias and spastic paraplegias, and to allow to profit from both of the expertise in both groups. And as you can see here, there is quite an overlap of the genes implicated in both groups, especially SPG7, as you know, or others. So when we did this, so my answer is yes to your question, Matthis. We have a network with 69 research and clinical teams. You can see on the right side the photograph of the first meeting. Paula Coutinho from Porto was there, and Ludger Scholz. You can see they're all quite older now, me included, but this was our first meeting. So then several other meetings came across because we wanted to share as a friendly group with minimal administrative restrictions that I have the privilege actually to share collaborations in. And you can see that there was a meeting in 2006 and 2009 in Paris. It's becoming a little bit bigger. The patient association were always very present at these meetings, and I'm very grateful to those because they helped us to have deeper insight in these patients. And in 2013, in our new institute, in the Paris Brain Institute in Paris, and 2019 in Nice, in conjunction with the Movement Disorder Society, which was the last meeting face-to-face. I hope we're going to do face-to-face meetings again. I would love to, at least, because I think it's a bit difficult to do it in Zoom still. What is SPATAX linked to? So SPATAX is integrated. It's hosted at the Paris Brain Institute, but it's based since the governmental plan in France to organize care in rare diseases. In 2014, the National Reference Centers for Rare Diseases were born, and you can see on the French map there that there are a lot of different centers in this reference, in the National Reference Center, which I'm coordinating. But this reference center is part of a larger group, which is called Brain Team, with 10 reference centers that are dedicated to rare neurological diseases, including 26 patient organizations again. So the link for the rare diseases between our expertise in the clinical and in the research domain and the patient organization is important in those. And we are all part of the European Reference Network, which is the overarched European organization led by Holm Krasner at the moment. And these are really more, not the SPATAX and the national reference centers are really doing the work on the ground, I must say, and the Brain Team and the European reference networks are more to advocate and to inform about the rare diseases and make things happen and possible. So do you have a registry? This was the second question. And is there a technical platform used for this? Yes, we have. It's based on REDCap, and it was started after a while after the start from SPATAX, because REDCap is not running since many times. So before we had Excel sheets like everybody else probably. And so we have now 6,000 patients in the network, but this is linked to a DNA and cell bank who has more samples. They're still not all in the REDCap SPATAX database. You can see an example here. If my mouse is working on the, can you see my mouse? No, probably not. Do you? No. But you can see in the different data collection instruments that they're where you can click, and then you have windows which are opening, but you can see this is an SPC7 patient who was followed in 2016 until 2020. So you can easily see the different details. You can change this and modulate. And it's, as Matthis said, for the ARCA database, it's web-based and you can access it wherever you are in the world. So what about the number of the autosomal recessive patients with an identified genetic cause or with a still unidentified genetic cause? And how many of these patients are actively seen or followed up? So what I can say for the moment, there are about 3,000 patients in the database, and as expected, because this is not a recruitment bias, there Friedreich's ataxia is the most frequent one this time, as you, Luiz, you said before. RFC1 expansions are looked for not for so long, so for the moment there are only 29 patients in the registry, and there are other, which I call AF-like cases, which are neither RFC1 or FX1, but they have the same phenotype, so it would be interesting to see what they are. In the spastic ataxia areas, there is ARCAs and SPG7. SPG7 is very frequent, I think, but there are a lot of other spastic ataxias. I don't know the gene. And you asked for the ataxia with ocular apraxia, so there are 42. And then we have a lot of dominant STAB1, not recessive ones. I have only 3 families of those with 2 mutations. But a lot of STAB1, more than 50, with a single autosomal dominant mutation, as we published recently. And we did a recent screen for CAPN1 and found quite a lot of them. Other genes identified in single families or very rare genes are another 60 patients. So you can see that about 20% have a genetic cause identified, but there are a lot of other families that don't have. I hope this answers your question, Matthis. What's about the recessive cases in this network? I wanted to show you this because when we did the study in recessive in about 300 cases with Marie Coutelier and Andy Singleton from the NIH on exome captured sequencing, it was interesting to see for me, and I just share this result with you because I think we have to integrate this in our search for new genes, that if you do a recessive analysis in exomes, we were able to identify causative variants in 23% with 74 patients out of this. But if you combine the analysis in also dominant with heterozygous mutations in dominant genes, you find more. You can add, we added at least 10 patients more, which made the final result of finding the causative gene to nearly 30% in this exome hunting study we did. And I think, which is even more interesting, that if you look at the clinical groups of these recessive ataxias we analyzed, the most frequent, or according to the different phenotype, we were able to identified the underlying gene, that in those which have a more late onset sporadic ataxias, for example, where we found very little genes implicated. So I just want to finish with this and show you the group. And like in all networks, you have to combine the research in the group that we lead together with Giovanni and Isabelle and Alexis Brais and Fred Darius and all those people, with the clinical part on the other side, which is our reference centers and the clinicians to be motivated to put stuff in the databases, which is important directly when you see the patient, so you don't need to upload stuff afterwards. So that's for SPATAX, and I hope that we're gonna do a lot of collaborative studies all together. Thank you very much.
Thank you, Professor Dürr, for this wonderful presentation, fantastic data from SPATAX. We are exactly in time, so the questions could be performed in the live room, coffee room. And now I would like to introduce Professor Bernard Brais, who is Professor of Neurology in McGill University, co-director of the Rare Neurological Disease Group of Montreal Neurological Institute and Hospital in Montreal, Canada, and who will present the topic ARCAs in Canada. Please, Professor Bernard Brais.
Ben, are you also still muted?
Mute. Am I better now?
Yes.
Yes.
Okay, great. Sorry for that. So I still wanted, for the people who know me, I'm a historian by training. So I wanted to say something about why ataxia, why Quebec has been a hotbed of ataxia research for decades now. And most of it stems from this guy, this amazing neurologist and researcher, André Barbeau, who was a close friend of many people in the 60s and a founder of neurogenetics worldwide, in fact. And he got very interested got very interested in ataxia and developed a very strong collaboration with a foundation and led by a patient. And they managed to raise millions of dollars to make Quebec the centerpiece of Friedreich's ataxia, but ataxia in general. That led to an amazing number of publications, over 300 publications during a period of about 10 years, including key publications on the diagnostic criteria of Friedreich, really a center that became important worldwide for ataxia research. Sadly, as I said earlier, Barbeau died in the 80s, and that changed, but he had a very talented student, or I say follower. He was not strictly a student, Jean-Pierre Bouchard, who described RSACs in 1978 in a preoccupation that was very close to the Quebec heart at the time, and still is, that we have regional founder effects. So he described from his region, Charlevoix, this spastic ataxia with a polyneuropathy. Most of the patients he followed are now followed by a physiatrist, Xavier Rodrigue, who partakes in our networking on RSACs. And then Jean-Pierre Bouchard just continued, and he's the one who really described the initial description for the SYN1, the ARCA1 cohort in the Brais area, and taken over now very much by Nicolas Dupré, who was involved in much of the process in Gibelot. So two of the common now recessive ataxia were described in Quebec because of founder effects and because of one guy who really had the eye and who was interested in ataxia research for over 40 years. So really it helps set the stage for what we're doing now. Because it has to be pan-Canadian, as you know, Canada is not a big country in terms of population, but really a big country in terms of territory. There's no ataxia center per se in the Atlantic provinces, Newfoundland, Nova Scotia, PI, and New Brunswick. There are very good neurologists who see those patients and will refer patients for consultation in Quebec and Ontario, but there's no clear center. Friedreich is the one associated mostly to this area. In Quebec, or central Canada, which is called Quebec and Ontario, the largest population base. The center founded by Jean-Pierre Bouchard and now taken over by Nicolas Dupré has a cohort of recessive ataxia. Those are data produced by Nicolas over a year ago. And as you can see, the center is really the ARCA center for ARCA1, SYN1 related recessive ataxias, and has very little follow-up, for example, of our ARC-SAX patients who are followed more in the rehab environment, but it has also a distribution of other recessive ataxias. I took this slide from him also. It just shows that despite the fact that we have often a common mutation that explains why some mutations are more common in Quebec, we have a lot of variability. I think we're up to over 20 mutations for RSACs in the French-Canadian population, but still one is the most important one that explains why it's more common in our population. Then a center that's very close to the heart of Matthis and I, the strong collaborator in the Prepare and now the ProSPACs, and led very much by Cynthia Gagnon, but who took over by Jean Mathieu, who is really the clinical expert on RSACs in many ways in the last few decades, and his young neurologist who's taking over, is the Saguenay-Lac-Saint-Jean Clinic, which has, as you can see, a huge contingency of patients, 162 living patients, but very little of the rest. It's essentially that ataxia. They have they've seen one Friedreich's ataxia case over all those years. If we go to the clinic in Montreal, Barbeau left a clinic that continues now to exist. I took over this clinic, in fact, from Massimo Pandolfo when he was director, when he was in his first Montreal stay. And so most of those patients are followed now in a rehab center, which I'm a senior neurologist, where we have a lot of RSACs, a lot of SPG7, a lot of ARCA1, some OAI2 also, and then it's more a sprinkling of many other things. If we go to the other descendancy of this large Barbeau clinic, is taken over by Antoine Duquette, and he's attending today, and he will answer for me the questions because unfortunately I have to test. He is really at this point very much responsible for the Friedreich's ataxia population. I've left him the entire population, so he's following all those patients from Montreal, and then he has a sprinkling of other ataxias, including the common one, ARCA1, in our population. Then if we turn to Ontario, Ontario is a huge province for us in Canadian perspective. Many of you in the paraparesis field will know the great work from Grace Yoon in Toronto, pediatric geneticist who follows over 300 children, or has seen over 300 children with ataxia phenotypes. And you see the distribution that she's provided. She's published extensively identifying new genes, and she offers really a very integrated multidisciplinary approach. So we have a referral center for pediatric ataxia, very strong one at the Kids in Toronto. We also have another city, smaller size, but a really well-known, internationally well-known clinician scientist, Mark Tapanowski, who follows some recessive and other neurogenetic disease. So he does have a sprinkling of the ones that maybe are more common, but also some rare forms. And then the other major adult center is in Toronto, where then many of you know Tony Lang, and then he has now Renato Monhoz and Elizabeth Snow, who follows their recessive cases. As you see, again, a sprinkling of cases. This is a very multi-ethnic population, so they have a little bit of everything. I realize that they have ADCK3 cases. I didn't realize that until recently. But really very strong collaborative and very much into dominant ataxias at this stage. Then in Ottawa, we have a superstar of genetic research worldwide, which is Kim Boycott, who developed Care for Rare, really major person, a major force in the introduction of whole exome sequencing in research, but in clinical practice. And she has a strong interest in ataxia and has a research clinical colleague who does the adult cases, Jody Warman in Ottawa, who follows again a sprinkling of cases, but not a large of series, but still does a lot of new gene research. So then if we go to Western Canada, this is even more difficult. In fact, extensive exploration on my part found only one young colleague who just came back from England who offers a more dedicated neurogenetics ataxia clinic, which is Dürr Pfeffer in Calgary, who sees also a sprinkling of cases without having a true series, extremely collaborative and wonderful colleague. And in fact, that's the end of the country. In Vancouver, they're seen by different people. There's no true ataxia clinic per se, at least not that I could find. So maybe I should end by saying that a lot of research and clinical care, but mostly research, is dependent on foundation and societies. We have 4 organizations who support in different ways: clinical services, research on ataxia. The oldest one is the one you recognize from the Barbeau period. It's now called Ataxia Canada, which is very FA focused. We have Muscular Dystrophy Canada, who does cover some ataxia, particularly ataxia with peripheral nerve involvement. And then the really leading foundation in terms of vision, in terms of for recent footprint, is the Ataxia Charlevoix-Saguenay Foundation, who really is a partner in all major initiative and has really allowed the research in ataxia, not only in Quebec, but elsewhere in the world, to really take a leap in the last 13, 14 years. So we have to thank them all for their support. So that's what I wanted to say to you. Thank you very much.
Thank you very much, Professor Bernard Brais, with a very beautiful historical note about ataxia, movement disorders, and epidemiological study on Canada. Thank you very much. Very beautiful history and great material. Anybody here would like to perform a question? I have a question from Luiz Leonardo Flores.
Mm-hmm.
Cases of mitochondrial ataxia are nuclear or mitochondrial DNA primary? Could you answer this question? It's not related to your point, but I think we have 4 minutes to talk about some questions.
I'm not sure if I'm the best one to answer that question. I mean, if the question is, should ARCA, for example, ARCA Global, be concerned about mitochondrial coded gene mutation that could cause ataxic phenotype? They're rare. I don't think that's the mandate, and maybe Matthis can answer that one. Clearly, some of the mitochondrial genes, or certainly a disease like ARSACs that has a mitochondrial component, are clearly under the umbrella of ARCA Global. So I think it depends on the genotype in a way, but certainly mitochondrial ataxias were not excluded at all from ARCA Global. And Matthis?
Sorry, I think finding his question in the chat, he was asking, it was specifically referring to the slides from Gray. You had a
We cannot hear you, Matthis.
Yeah, Matthis, all morning you've had a lot of cuts. We barely hear 50% of what you have to say. So I don't know, there's a technical bug.
It was just referring, he was just referring to the slide of Dr. Yoon. You said, so the mitochondrial ataxia nuclear or mtDNA?
I can't answer that question completely. I think she will, as a geneticist, it's all part of her. So whether she defined in her pie whether they were mitochondrial inherited or recessive forms, I honestly can't answer that one.
But I think
Grace.
The question could be also if Friedreich's ataxia is a mitochondrial disease, as we're going to hear later. So no, I think when I put mitochondrial, it's MT something. It's a mutation in one of the mitochondrial genes and not one of the nuclear encoded mitochondrial genes. Does this answer the question of the?
I think that's the better, stricter definition.
So let's move on for the next speaker. And now I would like to introduce Professor Brent Fogel, who is Associate Professor of Neurology of the Brain Research Institute, University of California, UCLA, Los Angeles, United States. And Professor Brent Fogel will present the topic ARCAs in USA, This is a pre-recorded presentation, but Professor Fogel will join us for to answer the questions and in the virtual room coffee.
Hello, and thank you for the opportunity to speak today. My name is Brent Fogel from the University of California, Los Angeles, in Departments of Neurology and Human Genetics. And I'm going to be talking today about the resources for autosomal recessive cerebellar ataxia in the United States. These are my disclosures. I have none relevant to this presentation. So there's an important need to have a robust network of centers for the study of autosomal recessive cerebellar ataxias. There's a number of different reasons for this. One is to enhance collaborative discovery of new genes, identify new mutations and such. Another is to characterize clinical phenotypes, perform natural history studies to better understand progression of disease. A third is to improve patient access to clinical research studies and to clinical trials. Things to study pathogenesis, biomarkers, and obviously drug development. And then lastly is clinical trial readiness. And this is one of the key aspects given the advances in developing drugs that can potentially treat different forms of cerebellar ataxia, and as we improve our ability to diagnose some of the rare genetic forms, improvements of other methods such as gene therapy. Now, in the United States, there is a precedent for developing a collaborative clinical research network in ataxia, and that is the Friedreich's Ataxia Research Alliance. And this is information provided to me from them. And this is showing the centers in the United States that participate in this network and their locations across the country. Now, we'd like to duplicate something like this for all autosomal recessive cerebellar ataxias. And in the initial phase, we have 6 preliminary network sites. And I'll tell you what those are in a moment. These are the locations on the map. We also have received interest from an additional 13 other sites around the United States, which we hope to engage and get involved in this network as well. I have data from the six initial sites, which I'm going to share with you today to give an idea about the number of patients this encompasses and what sort of information that we have at present. For these different endeavors. These are the sites: University of California, Los Angeles, University of Michigan, Emory University, Houston Methodist, Johns Hopkins University, and University of Pennsylvania. And now I'm going to go through the types of patients and diagnoses that are currently available at the different sites. Now, again, this data was collected all pre-COVID. Some of these numbers are higher right now, but haven't been fully updated yet. So this is just the minimum. So at the University of California, Los Angeles site, which is run by Susan Perlman and myself, we have a large number of patients with autosomal recessive cerebellar ataxias, including 165 patients with early onset ataxia, presumed to be recessive, and 386 sporadic onset cases, which could also potentially be due to recessive ataxia. About 30% of these cases in total have had clinical exome sequencing performed. There's 270 genetically confirmed cases. The predominant diagnosis of these is Friedreich ataxia, but there's also a significant population of patients with other forms of recessive ataxia, including mutations caused by the genes Senataxin, SPG7, RFC1, SIN1, GBE1, and others that are available for research and clinical trials. At the University of Michigan site, this data is provided by Vikram Shakatai and Peter Todd. There are 50 genetically confirmed cases. Again, the majority is Friedreich ataxia. However, there's also significant numbers of patients with SPG7, RFC1, Saxon, and other forms of recessive ataxia. Now, at the other sites, the patient populations are a bit smaller. The University of Pennsylvania, this data is provided by Pedro Gonzalez. There are 25 undiagnosed recessive ataxias, 14 genetically confirmed. The Friedreich ataxia patients are seen in a different clinic, so they're not included in these numbers. There are significant numbers of SPG7 patients, as well as the other conditions that we have talked about. The Johns Hopkins University site, this data is provided by Leanna Rosenthal. There are 31 undiagnosed patients with potential recessive inheritance, 7 genetically confirmed, with Friedreich's and RFC1 and ataxia telangiectasia predominating. At the Emory site, patients are provided, or the patient data provided by Chip Wilmot. There are 15 undiagnosed recessive patients, 40 genetically confirmed. Again, the predominant cases here are Friedreich's, but then there are others in the in the next tier of significant recessive ataxias. And finally, at the Houston Methodist site, data is provided by Tia Shizawa. There are 4 undiagnosed patients and 8 genetically confirmed. Include, again, predominantly Friedreich's, but then other rare ataxias as well. Now, in total, summing all of this data together, depending on whether we look at only early onset cases or include sporadic onset as well, there's between 240 and 460 undiagnosed patients with potential recessive cerebellar ataxia, and at least a quarter of them have already had exome sequencing that has been non-diagnostic, but still could potentially yield diagnostic information down the road. There's 389 genetically confirmed cases. I've broken these into tiers for what the most predominant conditions are. And again, Friedreich's ataxia predominates this. However, there's very significant numbers of other rare ataxias that, that ARCA Global is interested in, including Synofzik, SPG7, and RFC1, as well as notable numbers of patients with SIN1, RSACs, and GBE1 as well. Now, in terms of other data that's available across the sites, this is broken down in terms of whether or not DNA is available for additional analysis, whether all the patients have detailed phenotyping, whether longitudinal data is available over multiple visits, whether the patients have been seen within 2 years, and whether the patients have been consented to be recontacted for research studies. And in general, the majority of these sites have the majority of their patients available for that. The availability of DNA is somewhat limited at some of the sites, but those sites are working on developing protocols to collect more DNA of those patients for additional research endeavors. There is very high level of phenotyping across all of the sites, as well as longitudinal data. At almost all of the sites as well. And the majority of these patients are current and can be recontacted for future research. In terms of registries for contacting patients for particular clinical trials or research studies, at UCLA, there is the FARA registry, which is only for Friedreich ataxia, but then there's also the UCLA Ataxia Biobank, which does not contain any patients that have Friedreich ataxia, and all of those patients are available for potential research and clinical trials. Johns Hopkins also has their own registry within their ataxia center. University of Pennsylvania also maintains an internal registry as well of their recessive ataxia patients, whereas the other sites have no specific registry. In addition, there is the Coordination of Rare Disease, or CORDS registry at Sanford Research, which is partnered with the National Ataxia Foundation and provided this data on recessive ataxias. They have almost 2,000 patients enrolled in their registry, and almost 13% of those patients have a diagnosis before the age of 20. So if we use early onset as a surrogate for recessive ataxia, then there's approximately about 250 persons in that registry who have recessive ataxia. And the majority of the diagnoses are Friedreich ataxias, but there are also significant numbers of persons with potential diagnoses in Synofzik, aprataxin, Saxon, and others. So the future directions that, that we need to take with coordinating recessive ataxias in the United States, so we need to increase the number of participating sites. There are an additional 13 sites that we're interested in engaging, and we're hoping to do that within the coming year. Formalized procedures for collections of clinical data, including biospecimens and DNA, and that's already beginning and in early stages. Formalized procedures for collection of genetic data. There's also talks today talking about registries or repositories for this genetic data, including the centralized Ataxia Genomics Core and others. And improve patient access through registries and other means of accessing these clinical and research trials. And these efforts are underway. So I'd like to end here. I'd like to thank all of the centers that provided data for this presentation. Thank ARCA Global for the opportunity to speak, and I'm happy to take any questions that anyone has.
Thank you, Professor Brent Fogel. We have 5 minutes for questions and to discuss the last presentation. So I think Professor Matthis Sinovsky would like to perform a question for Brent Fogel.
Thank you. Yes. Excellent, Brent, you did an excellent job. I mean, from all over the US, but still the overall number of ARCA patients is so small. I mean, that is almost like in Germany, a small country like in Germany. So the hidden number of recessive ataxia patients in the US, which are not being captured by your centers. And even if you would enlarge your network by those 13 centers, probably, I don't know, they will have 10 to 20, maybe 30 patients each center. Still, it's a big number of recessive ataxia patients must be out there in the US not being captured. What is your estimate of that, and why does this happen? And can you comment on that?
That's a really great point. There could be a lot of reasons for that. It could be that they're not getting to ataxia centers. So we have a big country. We have a few ataxia centers. So they may not be getting there. They may be in movement disorder clinics. They may be in neuromuscular clinics. They may be in other neurology clinics and other places. Part of it will be reaching out to get those patients to us. There could also be there's a lot of patients who are out there that are sporadic, that aren't really necessarily categorized as recessive, that we probably should be capturing also. So, but they're definitely there. It's not like there's less.
Professor Fogel, I'd like to ask you a question. You showed some data with between 240 and more than 400 ARCA patients with undetermined genotype. And 25%, maybe around 25% you showed in this slide are have a negative whole exome sequencing. And what is your next step to determine the etiology of this case? Because I have some criticism, for example, for autoimmune ataxias, I think we should discuss this better, because maybe some forms classified as PACA, for example, could have a genetic etiology. Would you perform genome sequencing? What's your next step?
So we have a paper that came out last year that basically addressed this very question. It's and just to briefly discuss this for a second. So we've extensively evaluated. We've added evaluations for copy number variation. We've added analysis for repeat expansion to these cohorts, and we're able to up the number slightly, but we're still having a big gap in terms of diagnosis. So there's either things that we're missing, like genes that we're missing, which we know a lot of genes already, so there's certainly some out there, but that can't explain everything. There's got to also be types of mutations that we're missing and we're not capturing by exome sequencing. So there are other things that we're doing are genome sequencing, long read sequencing, evaluation for other types of mutation that aren't going to be seen by exome sequencing.
Great, thank you very much. And now, as next speaker, Professor Matthis would like to introduce me. Or should I introduce myself?
Oh, sorry. So Jose Luis is one of the, I think maybe the leading specialist from South America, and we met, I think it was the first time in person in Nice. So excellent just listening to you, how many patients you see, patients. So I think just in Germany we never see so many patients, work them up on such a so I'm really wanted to hear how many patients you see, which genotypes you see, and how you organize in America.
At the SPATAX meeting, exactly.
At movement disorders.
Ah.
Thank you. Can you hear me? I'd like to thank the organizing committee for having invited me for this presentation. I will present the ARCAs in South America. This is a great pleasure to introduce this topic here. And unfortunately, I do not have enough information from Central America. Although I have tried, I think we can have some information in the near future. So I'll focus my presentation on the ARCAs in South America. And I have nothing to disclose. And this is the outline of my presentation, introduction. I'd like to introduce the ataxia centers in South America, and I'll try to answer some points raised by Professor Sinovsky. How many autosomal recessive or early onset ataxia patients are there in each ataxia center in South America? What is the frequency of patients with a specific genotype? What types of characterization have been performed in how many patients of each cohort? What about longitudinal visits, cross-sectional visits, considering that Brazil, for example, is a large country? And what about the severity scales for ataxia, and about SARA scale and ICRAS scale? How many of these patients actively are seen and followed up during the last 24 months? And firstly, I'd like to start this presentation with this slide. I usually show this in my presentations about ataxia. We have to bear in mind that usually patients with autosomal recessive cerebellar ataxias present with a complex clinical phenotype. And the point is that we have to perform a deep phenotype, but we should not forget the genetic diagnosis. Professor Fogel showed the importance to have the genetic of our population, because we can offer a specific diagnosis for the family, for the patients. It's very important to investigate new genes, new cause of the disease. And also this is very important for research, for collaboration and future clinical trials, for example, considering gene therapy with the development of genetics in this day and age. So we have more than 250 gene mutations related to ARCAs. So I think it's very important to have genetic studies for these patients. And I'd like to introduce you the ataxia centers in South America. All these groups sent me information. I required information from all these countries. And in Brazil, we have 7 ataxia centers. Of course, we may have other centers that may care patients with ataxias, and they must be following in neuromuscular disorder centers or movement disorder centers. You can see that Brazil is a large country, and it's a problem because the vast majority of the ataxia centers are in the south of Brazil, and sometimes it's very difficult for the patients to move for an appointment in our ataxia unit. So there is a problem here to move to an appointment. This is why some patients have difficulty to longitudinal visits. But let's start with the number of autosomal recessive ataxias with early onset. We consider here patients that started ataxia before 50-year-old. I think we could miss some patients, for example, RFT1, but I try to consider the data that I collected from my friends. And this is the number of South America. This is a great number, a large number of ARCA patients. We have almost 1,500 patients with ARCA. And I'd like to introduce you the ataxia centers from these groups. And this is a very interesting meeting because we can know each other. This is a great opportunity to meet people and to understand what are the groups that care patients with ataxia. This is my group in Unifesp, Brazil. This is Professor Barsottini, who was my old mentor and the founder of ataxia unit in São Paulo, Brazil. And we have around 1,032 patients, maybe more, and 300 patients with early onset ataxia. This is my students, my PhD student, and my postdoc student that are performing this work to publish this data together with Matthis Sinovsky through ARCA Global project. We have around 300 patients in our unity, and we have 192 patients genotyped. You can see here the most common form, 72 cases of Friedreich ataxia, and AT 25 because we have a division, a pediatric division with a specific division for AT. RSACs, RFC1, maybe more. I think we may have more than 20 patients with RFC1 that are coming in the genetic lab. SIN1, AOA patients, AT-like, we forgot AT-like, Anotem mutations, the old ARCA3. Some patients with plan that present with ataxia, and remember Professor Dürr showed some patients that share some clinical features with the spastic paraplegia, the spastic ataxias. And I'd like to also to introduce my friend Professor Marcondes França Jr. from Campinas Unicamp, state of São Paulo, Brazil. They have in this group more than 200 patients with the recessive ataxia. And you can see here 29% of the patients with undetermined forms, and Friedreich ataxia is the most common, and some unusual forms. They are collecting data for RFC1, and I think it will grow soon. This is Professor Wilson Marques Jr. He participated on the first paper of RFC1 together with Professor Holden, more than 200 patients with recessive ataxias. And although they have a very important genetic lab, 70% of the patients have undetermined diagnosis. Professor Wilson is in Ribeirão Preto Universidade de São Paulo, Brazil, state of São Paulo, with more than 200 patients with ARCA. And in the south of Brazil, Professor Laura Jardim, who is attending this meeting as a chair. She is a specialist on SCA and a partner from our group with Rede Neurogenética in the south of Brazil, with more than 200 patients with ARCA in Porto Alegre. And note here, 73% with undetermined genotype. And this is Professor Eliotave, one of the pioneers of the study of ataxias in Brazil, the group of Curitiba, Paraná, Brazil, around 100 patients with ARCA and 44% with no genotype, undetermined forms, and Friedreich ataxia is the most common. You can see other unusual forms such as PNPLA6, GLUT1, and Marinesco-Sjogren syndrome. And I'd like to highlight these two groups because they are performing a great development in genetics and in the study of rare disease in Brazil, in South America. The group of Professor Pedro Braga Neto in north of Brazil, in Fortaleza, with more than 50 patients with ARCA. You can see here half of the patients with a no specific genotype. And also Professor Clécio Godeiro in north of Brazil, Natal, with more than 90 patients with ARCA. And note that almost 90% with a no specific diagnosis. And now I'd like to introduce you our brothers in Argentina, more than 100 patients with ARCA. This is Professor Marcelo Kaufmann, who is a leader in the neurogenetics studies in Argentina, particularly in the study of ataxias in the hospital Dr. Ramos Mejia. Professor Marcelo Kaufmann sent me this epidemiology from Argentina, Buenos Aires. Around half of the patients with a no specific genotype and 32% with a Friedreich ataxia, many unusual forms, but a small number of disease such as SCARB2, such as STUB1 and CTX, for example. And this is the group of Dr. Paula Safi. Here is her group in Santiago in Chile, with almost 30 patients with ataxia. I had the opportunity to be in Chile with her group, a fantastic group, and they have around half of the patients with a no specific genotype and around 30% of patients with Friedreich ataxia. And this is the group of Professor Mario Cornejo in Lima, Peru, around 50 patients with ARCA. Note here, around 80% with undetermined genotype, and most common forms are Friedreich ataxia. This is a summary of the characterization of the patients with ARCA in South America. I tried to include in this slide the clinical evaluation, frequency of visits, scales, and also the follow-up and DNA extracted. Our population depends on public health. All universities, I showed it to you, are related to public health, so it's very difficult to perform a genetic study. You may know that we depend on collaboration and we depend on funding. We are trying to improve our funding together with our partners in Brazil, with Professor Laura Jardim, Professor Marcondes, and Professor Wilson Marks. And anyway, we have to perform a deep phenotype in order to reduce the costs for genetic investigation. As for longitudinal visits, it's very heterogeneous because Brazil is a large country, and sometimes it's very difficult for patients to come to a visit, particularly in this pandemic moment. As for severity scale, I am trying to improve the use of SARA scale in our group, because it's very important for every visit of our patients, because we try to better understand the natural history of these patients. It's extremely relevant, and we are now trying to improve our genetic lab. You can see here Campinas, Ribeirão Preto, Porto Alegre, and this year we had the opportunity to have a funding, a governmental funding to perform whole exome sequencing in more than 100 patients with undetermined ataxia. So I think we are in a good moment to perform this collaboration with ARCA Global. So I'd like to show you my take-home message. South America has a large number of ARCA patients. This is very important for research and collaboration. There are several ataxia centers in South America. It was my great pleasure to present all these people, to introduce all these people to you. They are very important in collaboration. So I think we should keep this group together. And genetics of our population is poor studied. Several ARCA patients must still be genotyped, and this is a potential for discovery of new genes. And the ataxia centers in South America usually have a regular follow-up and include the minimal clinical assessments such as SARA scale. This is my group of fellows and Professor Barsottini. And my last message is that stronger we are, we do things better. So I think this is a great opportunity for collaboration with you. Thank you very much for the attention. I am available for questions.
Thank you very much, Jose Luis, for this comprehensive overview. Jose Luis, I think we have time for one more question. So there's one comment just by Luiz, but the comment, they have 70 patients from Mexico available. So, and I think, so you might want to join up to get some data from Mexico with him. That's just a comment, not a question.
Yes. Perfect. I'll contact him.
And a question from Paula. She's saying that is RFC1 sequencing in South America. Are you doing RFC1 sequencing in your lab, or who else is doing it in South America?
It's not sequencing.
Thank you.
Yes, Paula, thank you. I showed Paula group in Chile, and we are doing performing a project together with Professor Marcondes in Campinas and University of São Paulo, Ribeirão Preto. And this is performed in Professor Wilson, Ribeirão Preto, Brazil, the genetic testing. We are collecting the clinical data and in our group, but if you are interested in any test, in any genetic test, you can contact me.
And I have one a little bit worrisome that, can you understand me?
Yeah, there are some parts of...
Maybe my internet, sorry. Yeah. A lot of South American centers are not doing the SARA scale. And that is something, it really just costs 12 minutes. So that's not a problem of funding, I think. Do you have a perspective that this is more rolled out in other countries in South America as well?
Yes, I was very sad with this. And I think after questioning all groups, I think we can try to have a South America ARCA network in order to better evaluate this patient through SARA scale, ICARS scale. This is very important to better understand the natural history of ataxia. So I think it's mandatory. We should do this in all appointments of our patients, no doubt.
Okay, thank you, Jose Luis. So I can give the chair back to you and you introduce the next speaker. Thank you.
Thank you, Professor. And now for the next talk, I'd like to introduce Professor David Szmulewicz, who is Professor of Neurology and Head of the Balance Disorders Ataxia Service, the Royal Victorian Eye Hospital, Victoria, Australia. Professor David Szmulewicz will present the topic ARCAs in Australia. Please, Professor David.
Hello, everyone. I'm David Szmulewicz, a neurologist in Melbourne, Australia, and I'm going to be talking about ARCAs in Australia. I have tried wherever possible to use the structure supplied by the organising team. With regard to our own centre, we have a multidisciplinary cerebellar ataxia clinic in Melbourne. Traditionally, cerebellar clinical and research activity has centred around Melbourne, with a small scattering of clinicians in the other states. In Melbourne, we also have 2 neurogenetics clinics who consult on cerebellar patients as well as other potentially inherited neurological conditions. Our research activity includes deep phenotyping with an interest in natural history, speech, imaging, and gene discovery. We're also actively researching and interested in rehabilitation, as well as instrumented ataxia metrics. Unfortunately, we do not currently have a national network, but I will speak a little bit to that soon. With regards to the other states in Australia, Sydney in New South Wales, which is the other large city, tends to have their cerebellar patients seen in the context of neurotology, neuromuscular and movement disorders clinics. Perth in Western Australia has a neurogenetics clinic, which is the primary clinic seeing cerebellar patients. In Queensland, it is neurotology and movement disorders clinics, whilst the Northern Territory is a special case because of the Indigenous SCA3 communities, which are looked after by the Mikado Joseph Disease Foundation. At our clinic in Melbourne, we have dedicated investigation protocols, which include MRI, brain and spine scanning, which is carried out in collaboration with Ian Harding of Enigma Ataxia fame. We have speech research projects in collaboration with Adam Vogel, and have developed an ataxia neurophysiological protocol initially set up for deep phenotyping in Canvas, but is now applied to all our cerebellar patients. Our patients also undergo detailed central and peripheral oculomotor assessment, looking at objective oculomotor metrics as well as peripheral vestibular abnormalities, which are increasingly recognised in inherited ataxias. As mentioned, our clinic is currently planned to undergo an expansion to meet with a greater referral demand than expected. This clinic expansion, we also hope, will facilitate carrying out expanded research. We will accomplish this in part by increasing clinic frequency, but also by increasing staff. A part-time embedded research assistant will have the roles of coordination and research data gathering. In the past, our research collaborations have tended to be on an ad hoc basis from project to project, but our current aims include formalising and standardising research protocols, in part for patient enrolment into the ARCA Global project. With regard to the numbers of patients in our current state, being the past 2 years, we have approximately 60 patients with an identified autosomal recessive cause and approximately 50 patients whom do not have an identified genetic cause. As mentioned previously, we hope to identify many more patients, both prospectively and hopefully retrospectively, once we have increased our clinic capabilities and established a database. The numbers here do not include Friedreich's ataxia as per the ARCA inclusion policy, and it may be known by some of you that Melbourne has a large Friedreich's ataxia clinic with an international research reputation and is led by Martin Delaticki and Luiz Corbin. We're currently establishing a database, as mentioned, and in part leveraging off database of past genetic testing, that is, patients who were tested prior to the last 2 years. So the types of characterization that have been performed on patients has been quite variable historically, but in the past 2 years, approximately 95% of our patients have undergone deep phenotyping and 97% have had genetic testing carried out. 110 patients have undergone longitudinal visits with at least basic assessment on 2 visits, and a former 23 who have had lesser assessment to date with only cross-sectional visits and a basic assessment. Again, this is hoped to be significantly increased once we have established our database. Overall, in the past 2 years, approximately 150 patients have been consulted upon. Recontacting patients is possible, definitely in the case of those seen in the clinic over the past 2 years, and a lesser proportion from patients who were seen at multiple locations, but principally in the Melbourne-based neurogenetics clinic that I mentioned earlier. But this is something that we will be progressing again once the database is ready for us to populate. Geographically, our clinic in Melbourne has a statewide catchment, which is the state of Victoria. However, increasingly we're seeing patients from interstate, and we anticipate that this will increase over time as the clinic has a greater profile, and so do our research activities. Currently, the clinic we do not presently have a registry, but this would be part of the database we are establishing, such that both patients and clinicians would be able to enter data onto the database, and this could subsequently be interrogated for improvements in clinical service delivery and, of course, research data. The database will be set up in the REDCap application, and as I mentioned, this will be largely operated by a research assistant and a neurology trainee with oversight of one of the senior clinicians. Thank you for lending me your ears, and please be in contact if there are any questions. All the best.
We thank Professor David Szmulewicz. He's not here live. This is a pre-record presentation. We have no specific questions for this presentation, but I'd like to point a mistake. I'd like to apologize my friend Professor Christopher Gomez. In fact, we have the Pan-American Ataxia, hereditary ataxia, the PAHAM network, and this is a great opportunity to share with a North America group. And we had our last meeting in Havana last year. We were supposed to have this meeting this year in Brazil, but it was not possible because of the pandemic. But as I said, I was worried about the ARCA patients, and in this PAHAM network, we usually discuss the patient with SCAs, but I think we should integrate better our South America groups. So my apologies to Professor Gomez. I am part of the committee with Professor Laura Jardim, and this is a great opportunity to share this information in our network.
But Jose Luis, are those centers which are not doing the SARA, are they part of the PAHAN, or are they also not captured by the PAHAN?
No, the groups that participate on the PAHAN, they are ataxia centers that have a very important caring patients with ataxia, and then they perform SARA scale regularly, our group 2. I am worried because not all groups participating in South America study perform SARA scale. So I think we should share this information in order to better study natural history of these patients. As we have the PAHAN network, I think this is an opportunity to improve the collecting of data through severity of ataxia. So I think that we already have a structured network to improve the collection of this data in South America. So let's move on to the last talk. And lastly, but not less important, I'd like to introduce Professor Osamu Onodera, who is Professor of Neurology from the Division of Molecular Neuroscience in Niigata, Japan. Professor Osamu Onodera will present the topic ARCAs in Asia. This is a pre-recorded presentation, and there will be not questions after this talk, but we can discuss further questions in the virtual room coffee. So Professor Osamu Onodera.
Thank you for giving me opportunity to talk in this session. Today I would like to introduce recessive ataxia in Japan. Network for ataxias. For the second question, the number of recessive ataxia is very limited, and 1,000. And half of these individuals, the causative gene has not been identified. But the frequency of the recessive type ataxias, Asian population is very unique. There is no DNA diagnosis-free type ataxia in East Asian populations. The most frequently observed ataxia is AOA1, and AR6 and ABED, and very rare AOA2. To talk in this session. Today I would like to introduce recessive ataxia in Japan. But first, I would like to give the answer of the questions. For the first question, yes, Japan has nationwide research network for ataxias. For the second question, the number of recessive ataxia is very limited, and 1,000. And half of these individuals, the causative gene has not been identified. But the frequency of the recessive type ataxias, Asian population is very unique. There is no DNA diagnosis-free type ataxia in East Asian populations. The most frequently observed ataxia is AOA1, and AR6 and ABED, and very rare AOA2. And we have very unique system for the ataxias. All patients should visit hospital at least once a year. The hospital is a very specific type of the hospitals. And right now, the limited information is available from national database, but therefore, to accumulate the SARA or the other type of the scales, we have to re-evaluate the SARA for each individual. And again, all patients should visit a hospital at least once a year in Japan. Recontacting a patient is possible, and we have nationwide network. and each physician is well trained. The evaluation is performed by certified neurologists. And yes, we have a registry, but our registry system is very specific and has just started a web system. Before I start to talk about the ataxias, I would like to introduce an interactive and rare disease system in Japan. We defined these kind of diseases as interactive and rare disease. There are no clear cause and no established therapy are rare and requiring long-term care. For these individuals, we are eligible for medical fee assistance. For individuals there's a strong need for appropriate good quality medical treatment, we have a system to give them a medical fee assistance. For this system, we have a law, the law number Act number 50 of 2014, Act on Medical Care for Patients with Intractable Diseases. The object of this law is a medical fee assistance for patients of intractable rare disease, and also implement measures such as establishment of basic policies, promotion of surveys and research, and implementation of programs to improve the treatment and care environment. And for the budget, for the fund of the intractable diseases, we have two systems of the funding system in Japan. The one system was a clinical research. MHLW gave us a fund to research intractable diseases. The other is for the basic science and to find a new therapy for the intractable diseases. AMEDO gave us some kind of fund. Therefore, two different types of funding system exist in Japan to treat the intractable disease patient. This is a system of how to accumulate the intractable disease patient. The patient first visits the doctors, and the doctor gives them the application form. This patient is diagnosed as an intractable disease or not. And if this patient send this application to the public health centers, and after the evaluation by the specialist, the public health centers gave them the certification as intractable diseases. And then this public health center sent information to the prefectures, and the prefectures enter this information to the database. And MHLW has accumulated this database, and by using anonymous data, each researcher can reach the anonymous data. Again, the MHLW gives each physicians the nationwide standardized diagnosis criteria. Each patient is diagnosed by according to this clinical diagnosis criteria, and evaluate each patient and gives them the medical certification. And these individuals gave this application to the prefectures, and the another specialist evaluate the application and gave them the certification of the intractable disease. Therefore, two specialists, at least two specialists diagnose evaluate the diagnosis of each patient. And every year, this patient have to go to the physicians, and physician have to certify for each individual in every year. This is one of the form which accumulated by these systems. We accumulate this kind of the future of the diagnosis criteria application form, and therefore we can evaluate easily how many patients have these kind of symptoms in each type of the diseases. For the diagnosis criteria for ataxias, if each individual is diagnosed by DNA, or some of the families has already been diagnosed by the neuropathological findings, we designate each individual as a definite type of ataxias. But for the individuals who did not diagnose by genetic diagnosis has not been made, we designate this individual as probables. And the possible individuals that we cannot rule out secondary type of ataxias. After evaluation of these three types, we only including this system definite and probable individuals. For the QOL, we use modified ranking scales. We also accumulated the modified ranking scale for each individuals. For the individuals, routinely in Japan, the triplete type of disease, the DNA diagnosis has been made by commercially. But the other type of the diagnosis of the disease is very difficult. AOA1 and LSAX, the DNA diagnosis has been made in one institute, each one institute in Japan. But the individuals, this screening is negative. If the physician would like to do the next generation type of sequence, the physician can send the DNA to the JCAT, is a national system to diagnose SCA promptly. And the cost is totally free. Therefore, if each individual would like to clearly know the causative gene for each individual, they can send the DNA to the JCAT. By using these systems, we can estimate the number of the ARSCA in Japan. The number of ataxia patients in Japan, excluding MSSC, is 26,487. The percentage of SCA is 2%. Therefore, estimated number of the ARSCA in Japan is 550. As I told you previously, the frequency of the recessive ataxia is very rare in Asian populations. This is a comparison of the frequency of the recessive ataxia among the European individuals and also in Japan. In Japan, the frequency of the recessive ataxia is very low. The reason why the frequency of the recessive type ataxia is very rare in East Asian is the frequency of the Friedreich ataxia is very rare. We do not have a genetically diagnosed Friedreich ataxia from East Asian. Therefore, there's a big difference from the European population and East Asia. From the literature, in Japan, AOA1 is the most frequently observed type of the recessive ataxia. The next is ARSAX and AVED. And very rare is AOA2 is also observed. And also we have the AT, but mostly in the adult, it's very difficult to find the AT in Japan. And for the nationwide database, we have the frequency of the recessive type of ataxia. And for the individuals, the DNA diagnosed AOA1, the number of the DNA diagnosed AOA1 is estimated as 77. And this population is not diagnosed by DNA, but the clinical symptom is related to the AOA1, but we don't know what type of the mutations they have. And this individual has been diagnosed with AVED, therefore the nationwide, the number of the AVED should be estimated at 22. Our institute performs the DNA diagnosis of the AOA1. By far, we accumulated 39 families throughout Japan. Therefore, maybe more than 50% of the individuals who suffered from the AOA1, the deeply clinical symptoms has been accumulated in our institute. This is the frequency of the mutation, and very interestingly, there's a tight founder effect exists in Japan. Only 1, 2, 3, 4 type of the mutation has been identified in Japan. For the ARCAs, the Yamanashi University performed the DNA diagnosis for ARCAs in Japan. They accumulated at least 17 families throughout Japan. Therefore, also the maybe most of the ARCAs patient clinical information has already been accumulated in Japan. Speculated. Again, I answered the question of the for these meetings, and yes, we have a nationwide research network for ataxias, and the number of ataxias is very limited, as I told, and at least half of them has not been identified the cause of the gene. The frequency of the ARSCA in Japan is AOI is most frequently observed, but ARSAX and AVED also observed in Japan. And the unique part in Japan, all ataxia patients should visit a hospital at least once per year and perform the clinical evaluation by the neurologist. Therefore, we can accumulate a detailed scale for all of the individuals if we would like to do. And however, right now, the limited information is available from national database. We have to re-evaluate the individual for the thorough. And also, all patients should visit the hospital at least once a year. We contact the patient possible, and each clinician, the physician, is well trained. Most of them is a neurological specialist. And all the system has just started, but this system is limited. It's undiagnosed individuals. Therefore, as a registry system, we have to rethinking the other type of system. Thank you for your attention.